Okay, well, thank you, Matteo. As you said, I'm Dennis Montoya. I'm a computational immunologist in Matteo's lab. And today I'll be presenting a tool that we've developed, CELLFI, which is able to determine the cellular epigenetic fingerprint, which can be used for the computational reconstruction of immune cellular microenvironment in tissues. So a common research approach in differentiating two different disease states is to take samples, maybe blood samples or patient biopsies from cases and controls within different patients, extract the RNA or the DNA, and then perform a transcriptome or methylome DNA methylation analysis. You average the case and controls, do differential expression or differential methylation, and then through that, look for pathway identification, diagnostic markers for the idea of developing new clinical tools and therapies. However, what's been increasingly clear is that these different clinical samples that we have are not homogeneous, singular tissues. They actually are composed of a number of different cell types. And you could think of each cell within the tissue of having its own methylation, methylome, or transcriptome. And so you have to be able to differentiate that between the different immune pathways that might be within these patients. And then similarly, there's also patient variation between each of these samples. And so the Pellegrini lab, we've developed previous tools showing RNA, using RNA transcriptomics to look at these different pathways and actually output cell frequency or cell activation patterns on a patient-by-patient basis through Savant, and then GEDIT you'll actually hear about in the next session from Brian from our lab. However, for today, what I'm going to be focusing on is this deconvolution of the different cell components based on DNA or DNA methylation. And here, it's the main idea is that we want to be able to use this cell information to correlate with some sort of disease risk or disease trait, because it's become increasingly clear that the type of cells that are present and the magnitude at which they're present within the patient lesion often correlates with the prognosis or potential therapy success within patients. So just a little quick background on DNA methylation. So here we look at CpG methylation. And so DNA methylation occurs at these CpG dinucleotides. So if you look across the sequence of DNA, we're talking in particular to when there's a C next to a G. The P is for the phosphate, which is in between here. And then there's a methyl group, which is attached to this cytosine. And so the effect of this is that it can turn regulatory elements such as promoters. The dogma is basically that when something is methylated, it tends to be more inactive, more heterochromatin, whereas when it is unmethylated, it tends to be more active. And so in terms of the immune system, which is derived all from the hemopoietic stem cells, so this is hematopoiesis, going from the stem cell all the way down to the different immune cell components. As you go through the development, you start off with a cell that's probably mostly demethylated and pluripotent to these terminally differentiated cells. And during this process, basically, if you're a B cell, you're going to want to turn off all the other cell type genes and loci, so then the DNA methylation actually increases in those regulatory regions which are unrelated to B cells. However, those regulatory regions which are in B cells will become demethylated because you want to be able to turn on B cell genes. And so DNA methylation dynamics actually help determine the cellular identity during cellular development. And so can we use and exploit this DNA methylation developmental differences to quantitate cell frequency? And so as a proof of concept, Eugene Hausmann has done this using DNA methylation arrays. He's shown here are purified cell types. So each column here represents a different purified cell type. and the rows represent the amount of CpG methylation, with yellow being fully methylated, so 100% of those is methylated, versus blue is not methylated. And so what you can see is that there's very different patterns between the different cell types as expected and shown by other groups. But what he did was he selected the most different CpG loci between these cell types using a particular method, mainly using ANOVA, and then used these sites in a mixture of known concentrations. So each one of these is a different mixture, and then in here is the percentage of the different cell types. And here are the expected values and the observed values, and so they tightly correlate. And since this, they've actually improved it quite a bit. However, some of the limitations of this study is that it uses array data. So array data are these microarrays for methylation, the CpG methylation, and has limited genomic coverage. So, I mean, this is also increasing. Started off with 27K, then 450K, now they have 850K. Whereas sequencing can provide much more CpG loci to begin with. And so the technique that I'll be using a lot here today is reduced representation by sulfite sequencing, which focuses more on CpG islands and gives you, anywhere between 1.5 million to maybe up to 4 million CpG loci. Whereas if you use whole genome bisulfite sequencing, you can get greater than 20 million if you want to pay for that. And as we've come to see, is that sequencing is becoming. We've already passed the point where sequencing is often cheaper than microarrays for various samples, and so this is the case for this DNA methylation sequencing. RRBS is about $150 per sample. This is including both the library prep and the sequencing cost. Whereas the methylation EPIC chip, just the retail price from Illumina is about $400 per sample, plus you have to pay for the actual running of the microarray. The other thing is that there was a limited number of cell types that were tested here. And so with one of the graduate students in the lab, he helped kind of bring this tool to life and programmed a Python script called CELLFI. And so CELLFI is short for Cell Epigenetic Fingerprinter. And what it does is it starts with reference methylomes from purified cell types. And most of the studies that I'll be discussing here are from the Blueprint Epigenome project, which uses whole genome bisulfite sequencing. And then you have your samples or your mixtures, which we've usually used reduced representation bisulfite sequencing. So the first step is very computationally heavy. In that first we have to, so the difference between microarrays and sequencing is that the coverage of microarrays is the same every time. You always get the same 450K CpG loci, but with sequencing, it'll vary depending on the run. So we have to kind of combine all these samples with their various algorithms over the millions of different sites. And then we add another thing, which isn't available in microarrays oftentimes, is that we actually are able to cluster the regions between different CpG loci. So the CpG loci across the genome tend to correlate with the run that's next to it. You get a lot of runs where there's a lot of methylated CpG loci, and then other runs where it's demethylated. So we take advantage of this and incorporate these different regions into loci. So the first thing we do after putting together all the data is to then select certain regions where the CpG methylation is correlated. Next, what we do is to then do the epigenetic fingerprinting. So with different algorithms, we identify CpG loci which are most characteristic of a certain cell type, usually demethylated. So in this case, if this is a mixture, you can see that we've selected region A because region A is demethylated here in this B cell and methylated in T cells and hopefully the other cell types as well. Whereas we have another region which is demethylated in T cells, but then methylated in B cells. And so when we look at our mixture, if we only look at these CpG sites, we can then infer the quantitation from that. So we then merge these selected CpG sites with the sample mixtures to then estimate the cellular fractions through linear regression. And so some of the applications for this is obviously cell quantitation, which I'll be focusing on today. You could also use cell type of origin determination, like for cell-free DNA. If you have a piece of DNA with a certain methylation pattern, this may be able to help determine what type of cell that came from. Developmental analysis between cell types. So, these are probably going to be functionally important for the cell, these certain selected loci. And then possibly also for EWAS correction. But I think, there's other methods, reference-free methods, which probably our last speaker, Aaron Halperin, may cover. And so this is our selected CPG sites that we've done through CELLFI using the Blueprint Epigenome project. And so what you can see is that the ones in blue are the ones that are demethylated. And so these are a set of loci that are demethylated only in B cells or NK cells and T cells. And so we do that for all of them. We also have certain loci which are different just in these CD4 versus CD8 because they're very developmentally similar types of cells. They're both T cells. So we focus in on some regions that are particularly different from that. And so kind of looking at one of these regions. So each one of these represents the reads that we see in our purified cell references and then our in vitro genome mixtures. So in this region, so these same 3 CPG nucleotides are from the same region for all these different descriptions here. And what you can see is that this region happens to be highly demethylated within the monocyte, whereas it's more hypermethylated in every other cell type. And these are the types of regions that we'll select with CELLFI. Now when we look at the in vitro known mixtures, which I'll present later, we see that depending on the amount of monocytes, so like this first mixture has a lot of monocytes, has a high demethylated aspect, similar to the monocyte here, where ones with fewer amounts of monocytes have a lot less. And so this is kind of the basic concept of how we're able to estimate the frequency for that. And so using these known mixtures, so this is where I purified cells from a donor here at UCLA. I purified each of the cells and mixed them at a known concentration. And just kind of as a validation set, this was done with blood from an entirely different country. We sequenced it by RBS instead of whole genome by sulfide sequencing, different lab processing. So this is really an external validation set. And what we get is pretty tight correlation between the amount of the CELLFI estimate compared to the actual cell frequency, with an average accuracy of about 4% across all the different cell types. We also have done this in the microarray platform as well, such as originally what Houseman does here is the more Houseman technique implemented through MinFi, which is a popular R package, bioconductor package used for DNA methylation microarray analysis from Raphael Harry's lab, which has a decent amount of correlation here, but the CELLFI seems to outperform that analysis. Furthermore, Shashank Najahara, one of my undergraduates, developed a web application for these Illumina methylation arrays. Because Illumina, again, like these microarrays are much an easier process because they have the same covered CPG loci. So using the pre-selected CPG loci now, we've developed a web application where you can just upload your microarray data, so just your probes and then your methylation beta values for your samples. And then within less than a minute, we'll be able to give you the actual estimates for all your different cell types here. This example is showing colon cancer samples, either the cancer or adjacent normal samples, and you can see that the tumor cell count is higher in the cancer samples than the adjacent normal. And so another application that we wanted to try in terms of disease is in severe combined immunodeficiency disease patients. These are patients that are born without an immune system. This is the boy in the bubble syndrome that you may have heard of earlier. Basically, these patients don't have an immune system, but there's recent studies in Donald Cohn's lab, which they actually do gene therapy. So you get the donor stem cells, do gene therapy to repair the mutation, and then reconstitute the immune system within the patient. So normally these patients don't have any lymphocytes or immune cell or other types of immune cells, and then Afterwards, they measure the amount of lymphocytes to see how well are they reconstituting their immune system. So we got 5 patients of those, and we have both the clinically measured value, and then we compare that with the CELLFI estimation. And so what we could see is that there's a very tight correlation, again, where CELLFI estimates very well the amount of lymphocytes and other cells within these diseased patients. However, CELLFI can do more than just these standard cell types. We can do other cell types as well. And so because the immune cells are becoming reconstituted, they're at a very lower developmental state. And so one of these, there's many stages in terms of the neutrophil development. And so one of the pre-cells is this band neutrophil before you get the mature neutrophil. And so we wanted to see, well, can we see some more of these immature cell types within these patients? And so these are the different patients. Number 1 is a healthy patient, and then the rest are the different rankings as explained by the clinician. We could see that the lymphocytes, are higher in those that had a better outcome than the ones that had lower lymphocytes. But interesting, these band cell neutrophils seem to have the opposite correlation. So there seems to be more of an immature developed immune system in these patients with poor outcome. And so but we don't see that with the monocytes. And so this is something where CELLFI actually added some extra value that wasn't previously known with these patients. And so we have a number of collaborations applying CELLFI to disease samples, which I won't have time to show these studies, but they're very interesting in terms of metabolic syndrome, done in adipose tissue, tuberculosis, colon cancer, heart disease, and the list is growing and growing. And if you're interested in collaborating, definitely email me at this email address or talk with Dr. Pellegrini as well. And so with that, I would like to give acknowledgments. Obviously, Matteo was definitely helpful in kind of giving me the tools to pursue this idea when I first brought it to him. And Larry Lamb also is a graduate student who really brought it to life in constructing the full version, which is a Python script of CELLFI, which works well over Hoffman cluster. Upon publication, we'll be releasing this on GitHub as well as the Illumina web application. It'll probably be listed in the Pellegrini Lab resources as well. I would also like to thank some of my undergraduate students, Shashank, who did the web app, Jim, who did some of the mapping, Gerardo, who did a lot of the region visualization, and Frida, who helped with some of the earlier simulation data. And again, if you want to use the beta version of a lot of these samples, please contact me. We can work something out. Thank you.